Okay. So we're characterizing the microbiome of developing floral tissues. So the microbiome is defined as a collection of microorganisms inhabiting an environment. And by microorganisms, I mean fungi, yeast, and bacteria. So now, the interesting thing is when you think about raspberry fruit, it really starts at bud break, the whole developmental process. And the morphogenesis from buds to flowers to fruit is a very dynamic process. You can imagine you start with just emerging new tissues, and then you form a flower. And on the flower, it's covered with nutrient-rich stigmas. And then the plant begins to change, and you get to the green fruit, the immature fruit stage. Chemically very different for those inhabitants on the surface. And then finally you get to ripe fruit. And again, that's very chemically different from all the other stages. So it's a very dynamic tissue system. When you think about it, microbes have to colonize and grow on these changing tissues while exposed to fluctuating conditions like rainfall, intense sun, extreme temperatures, warm temperatures and cold temperatures at night, insect visitors that might bring in new microbes, and also other microbes that are already residing and possibly competing for nutrients and sites for growth. So one of the questions I'm interested in is, do the early colonists on the early tissues persist throughout this whole developmental process? And which are organisms favored by certain stages of development? So do we find certain things on certain tissues and other things on different tissues, or is it fairly consistent? In the long term, we want to understand the microbiome because this may impact disease processes. So to study the microbiome, we use a two-pronged approach, a molecular culture-independent characterization of the microbiome. So essentially, we freeze-dry tissues, we extract DNA, we grind the tissue, the freeze-dried tissue, we extract the DNA, and then we use PCR to amplify universal genes for bacteria, it's called 16S if you've heard of that, and fungi and yeast, the ITS region. And then we analyze the sequence and identify the genera of the microorganisms present and their relative abundance. So the advantage of this system is that you can detect all DNA from all the organisms that were present on these tissues in one fell swoop. The disadvantage is that sometimes you get uneven amplification or representation of the genes just because of the variation in the gene sequence. Also, dead cells may be amplified because the DNA was extracted from dead cells that persist for a while. And also sometimes the databases to identify the sequence is limited. Anyway, it's a very powerful approach to look at what organisms are present in a tissue. The other approach is culture-based characterization, and this you might be a little bit more familiar with. Essentially, this is what we've been able to accomplish so far. We match tissues in buffer, which I'll show you in a few minutes. We spread the washes and dilutions onto culture media, such as tryptic soy agar with cyclohexamide, and that permits the growth of bacteria. Or else we use PDA with streptomycin, and that encourages growth of fungi and yeast. We then look at these plates, we record the numbers of organisms that grew and what they look like, the morphology of the organisms recovered. And then we purify and store the cultures, and we identify the organisms by morphology and molecular assays. So there's some challenges to this method also, because not all organisms can grow on those two media. There's many organisms that would be considered non-culturable or unable to grow on those two media specifically. Also, sometimes you can get a fast-growing, aggressive, like fungus. They'll just grow over the whole plate and obscure the other organisms. So sometimes there are some biases, but the advantage of this method is that you get isolates, which you can use in downstream studies. So this is what we set up this spring. We worked in the Linden area in red raspberry fields. We established sites in 3 meeker fields and 2 Wakefield fields. And then we set up 5 replicate blocks per field, and just random, they were scattered throughout the field, and they were between 2 posts, so they're like 10-foot long rows. And then we went ahead and periodically sampled them. We took 10 samples, tissue samples per block. To transport them back to the lab, we put them in sterile dishes like this, or with larger fruit, we put them in large plastic boxes. So the stages that we sampled were bud break, when the floral buds just emerge, then also pre-bloom, so you start to get some white on the petals, but the flowers aren't open yet, full bloom, green fruit, and then ripe fruit. And these are commercial fields, so these first two samples were not exposed to fungicides. Sprays started right after that pre-bloom stage. So then we take the samples and we had to process them. So essentially we go from here to here onto culture media. So we had two methods. One was direct plating of samples, and thereby we took the samples, put them in little plastic bags with some buffer to support the microorganisms in there, and we mashed it thoroughly by hand. And then we took those suspensions of tissues and put them in, transferred into test tubes, and then spread them onto media, such as this. The other thing we did on just fruit was surface disinfestation before plating of samples. So we took our fruit, we washed them, we soaked them in bleach, a dilute bleach solution, and then ethanol, and then we rinsed them with water. And then again, we put them in, transferred them in a sterile hood into bags, mashed them, and similarly put them onto plates. So what did we get? This is I know it's a very busy graph, but I'm gonna walk you through it. So this is the average incidence of detection and the population sizes in Meeker raspberry over time. So the top graph is what proportion of samples from a field had detectable microorganisms. And the microorganisms are indicated by the bar color. So bacteria are indicated by blue, red is fungi, and green is yeast. So for example, in site 1, we found that 40% of the samples or so had culturable bacteria, and then 20% had culturable yeast, fungi, and about the same for yeast. And you can see that over time, the incidence of detection increased and was especially high for bacteria at full bloom, which is this stage right here. On these graphs, it's this stage. We also monitored the population size, and that's presented here. And this is a logarithmic scale. So 2 means 100 cells, 3 means 1,000, and so on, 10-fold increase. So we see the populations are fairly stable, except at full bloom, we see a spike in the bacterial population sizes. The interesting thing to me is that during green fruit and ripe fruit development, we really had high populations of all 3 types of microorganisms on these samples. This had the lowest incidence of detection, but between 90 to 60% of the samples had bacteria, between 60 and 70% had fungi, and almost every sample had yeast. And we see in these other 2 Meeker fields similar patterns where almost everything had culturable bacteria, yeast, or fungi. Looking at the Wake field, here again, this is between bud break and full bloom. We didn't get a pre-bloom sample from these fields, but again, we find a fair number of microorganisms right at bud break. So when the tissues are just emerging. At full bloom, again, we had a high incidence of bacteria, and then at green fruit and ripe fruit, we had high populations of all types of, high incidence of colonization by all types of organisms. The population sizes weren't that large. They're only about 100 to 1,000 cells, but almost every fruit was colonized. And again, remember that here we're finding fungi and yeast, even though these fields have been sprayed since before full bloom. So I found that very interesting. Now, so let's take a look at some of these bacteria. Who's out there like at full bloom? So these are across all 5 sites at full bloom, and we just took bacteria from the various plates and spotted them onto new media. And you can see that, hopefully you can see that the morphology and the color of the colonies is really extremely variable amongst all these samples. So there's a lot of diversity with the bacteria found on red raspberry fruit flowers. Now we're just going to take a look over time. So we have all this diversity at full bloom. Do we have diversity at earlier samples and also from fruit? So looking over time, here's bud break in one site on Meeker. You can see here there's pinks, there's yellows, there's white colonies, all sorts of different colors. We also have those at pre-bloom. At full bloom, it starts to look a little bit more homogeneous. And then at green fruit and ripe fruit, it looks all pretty much the same. We've done some identification of these bacteria. And if it comes up, there we go. So preliminary identification, we find Pantoia glomerans. It's one of the yellow bacteria. That was interesting to me because that's a genera used for biological control of fire blight. So there's known to be biocontrol agents in that family. Pseudomonas species, Citrobacter, Rotella, Rosenbergiella, Pentabacillus, and Cyclobacillus. And then of course we have some of the uncultured bacteria or unidentified bacteria in the databases. These are done by molecular analysis. But here we have just a wide assortment of common environmental bacteria colonizing up through full bloom. Now the major organism at late season was Bacillus. And that's what you see on these green fruit and ripe fruits. And we think that these Bacillus is very tolerant to the drying conditions that you might have out in the field during the early summer months. Interestingly, actually that bacteria in the Bacillus was identified as Bacillus thuringiensis, which you might know as BT. I don't know if anybody was using BT, and I don't know if this is the commercial organism, but it was a member of that species, as far as we know. We're going to be doing some more tests, but it was interesting that we were finding pretty homogeneous identification of Bacillus in these fields during on the fruit. Now, the question is, are these organisms inside or outside the fruit? So here we surface disinfested the fruit, and we compared the bacteria that we got from those versus direct plated fruit. So on direct plated fruit, we see a fair number of different organisms from these samples. When we surface and disinfest, generally the populations are down, but not completely wiped out. We don't think that these bacteria are actually inside the fruit. They may be, but it also may be that they're residing in biofilms that are resistant to some of the treatments we applied. But we certainly say that when we did the plating, there was a much, much higher population on direct plated fruit versus surface sterilized fruit. So we think that most of the bacteria are residing on the surface of these fruit tissues during development. Okay, let's take a look at yeast. These are the yeast present during full bloom across all 5 sites. And again, just like the bacteria, we do have color variations. And again, the question is, were these yeasts that were present at full bloom, present early and also late? Ooh, running out of time. So this again is similar to the last story where we see a lot of diversity during these bloom stages, and then it becomes much more homogeneous on the fruit. And here we find primarily Cryptococcus, Sporobolomycetes, and the major yeast present is Oreobasidium pullens, which is the spreading yeast on these plates right here. And that is related to, that's the same genera and species that's found in the material Botector, which is interesting. And Botector is shown to be a non-fermentive yeast. So these yeasts might be present on fruit, but they're not, presumably non-fermentive. If we look at where the yeasts are present, here again we had yeast from non-surface sterilized fruit versus surface sterilized, and we see that most of the yeast during surface sterilization are killed. So the yeast are pretty much on the surface of the fruit, not internal in the tissues. Okay, let's look at fungi briefly. Here's an example of some of the array of fungi that we find on tissues during pre-bloom. Again, very diverse. We have this same story. We stacked them into different categories, And you can see that we have really high stacks of many different categories during all these early bloom stages. And at green fruit and ripe fruit, the diversity drops. And again, the preliminary identification of the fungi is alternaria species, Botrytis, Diporthy, which is Phomopsis, and Penicillium. Specifically, I just want to take a second and talk about Botrytis. We did find Botrytis on the fruit. We also isolated early in the season from three of the five sites by pre-bloom, and at the fourth site by 10% bloom. Now, the incidence of Botrytis species on pre-bloom tissues was really low. It's about 2 to 6% of the samples. And we did not determine if early season isolation of Botrytis represented just Canidia landing on those tissues that we sampled, or if the tissues were really infected. Olga's working on that, so she'll be able to tell you that at some point in the future. That's outside the scope of my study. The mean incidence of storage rots by site, at the end of near harvest, we went ahead and collected 50 additional ripe fruits from each block. And then we surface disinfested and stored them in humid chambers. And then we determined the incidence of fruit rot over 12 days. And we can see that within the sites, we do get variation in the incidence of fruit rot. Most of these rots were due to Botrytis species. And then the later rots that occurred over the 12 days were generally associated with alternaria. So we did find a lot of diversity in the incidence of disease here. So essentially, we showed that microbial communities really do change over time and over different developmental stages of fruit development. And by direct plating methods, we isolated the greatest diversity of bacteria, yeast, and fungi from floral tissues. We isolated bacteria such as Bacillus species, yeast, Oreobasidium, and fungi from nearly every green fruit and ripe fruit. So the diversity of culturable organisms decreased on fruit compared to bloom samples. And we're hoping that the studies of the microbiome will provide insight into the interactions between the microorganisms and the microbes, between microorganisms on these tissues, and also microbes with plant tissues that may influence the disease process. So stay tuned, we'll have more information next year. I'd like to extend my sincere thanks to the Washington Raspberry Commission for providing funding. The cooperating growers, I really enjoyed the field visits and the chats, learning about the crops. USDA ARS Crops Research Unit, and the members of my lab, Gail McGee, Brenda Schaefer, Rochelle Castro, Brenda Villanueva, and Ching Yang. Thank you.
Right, any questions?
Oh, questions, okay. Pete, or I'll take Pete first.
So I'm wondering, you didn't have any samples for any untreated, but I'm just really curious if there would be a singular species of bacteria and or fungi that would take over an untreated that's dominant out there, and what role that might play in even commercial blocks.
So we did not have any untreated blocks. It's a good question. I didn't see much evidence that the fungicides were actually affecting bacteria per se. I think that probably a change in the environmental conditions were a bigger factor. And I think that what happens is, when we're doing biological control, we normally flood a system with a single organism and get it established and excludes everybody else. But here it looks like there's so many stigmas and so many sites for these organisms to colonize that you do get this whole community structure occurring under natural conditions. So I think that competition just kind of keeps everything in check, and until the conditions are unfavorable to like Pseudomonas, then Bacillus will take over eventually under those tough conditions. Does that make sense? Yeah. It looked like Bacillus, and I know Oreobasidium is very present. In talking to colleagues that work on pear, they find that Oreobasidium's all over the place in pear and apple. So it's a good colonist of aerial tissues. Tobin.
I was just wondering, have you screened any of the fungi that you've recovered for sensitivity to fungicides? Because I'm surprised, if these fields are intensively treated with fungicides, you have natural resistance or.
Yeah, I've been curious about that. So some of the, like alternarias, I did do some screening for fungicide resistance. Some of the materials, like the alternarias that you would study for resistance at concentrations that would inhibit botrytis, they had no trouble growing on everything. So they were very resistant at that concentration. But I haven't looked at what the effective dose is to inhibit some of those organisms. And then we did look at early season botrytis. The few samples that we had, they're resistant to fenhexamide and boscalid, but nothing else. And then going back to the same fields later on, we had isolates that were sensitive to fenhexamide, and some that were sensitive to boscalid, and some that were resistant to cyprodionil. So it really did shift over the season, and hopefully the research that Olga's doing will be able to address some of those questions. Yeah. No, botrytis wasn't the most common fungus. We're still working on that, but there's a lot of different fungi. At the end of the season, we did have botrytis in surface sterilized fruit, but suggesting it was inside the tissues. And we also had alternaria, but on non-sterilized fruit, just native fruit, it was, as an old colleague of mine used to say, dog's breakfast. So it was just about everything. Okay.
All right, I think we're going to move on to the next speaker.